Thank you. All right, we ready? So hopefully you guys have had a chance to already look at or watch the other videos that I put up, or at least most of you have. Because I'm going to kind of assume you've already seen those as I move on here. So I might talk about some things that we've seen in the earlier presentations. But this is the first live lecture I could get to do in the genetics section. Next week, Dr. Gardner is going to be here. Once again, I sat in on his lectures last year. I thought he was outstanding, so I think you guys will really enjoy him. He's got a little bit different approach than I do, but he's got a really extensive knowledge of genetics, so he's a good source. Anyway, so this autosomal inheritance lecture set was actually 2 lectures, and I roughly divided it in between discussing autosomal dominant and autosomal recessive diseases. And we talked about the autosomal dominant diseases in the last lecture. So today we're going to move on and talk about the autosomal recessive diseases. And then we're going to do at the end a little bit of population genetics, a little bit of math. And I promise you, if you hang in with me with the math stuff, I'm going to make it easier by the end. And it's fairly simple math, although it is math, and it is something you're going to be required to do. Okay, so now we're going to look at some autosomal recessive conditions. One of the things I want you guys to be able to do is see a pedigree and recognize which pedigree we're talking about. Now, at this point, this is only the second type of pedigree we've seen. Tomorrow, I'm going to finish the 4 main types of pedigrees. And when I get through all 4 of them, I'm going to explain to you how I look at a pedigree, the logic I use to look at a pedigree and determine what it is. If that works for you, great. If you have a different way, that's fine. And I'll also give you a little figure that you can also use that figure if it helps you to kind of eliminate one pedigree and narrow it down. But one of the things you do want to be able to do is recognize a pedigree. So we're talking about autosomal recessive. So for an autosomal recessive disease, 2 mutant alleles must be present for the disease to be expressed. So with the dominant, you only needed 1. With autosomal recessive, we have to have 2 mutant alleles. Okay? So these, and remember how to read these pedigrees. So these are affected individuals. They have 2 mutant alleles. An affected person is usually born to unaffected parents. So with autosomal dominant, you always had an affected parent unless you were the first person to show that disease, unless you were the proband or the new mutation in the pedigree, you always had an affected parent. Either sex can be affected. Now we're going to use that later on to differentiate between some other pedigrees. Both parents of a child are obligate carriers. That means, so if we're looking at this pedigree, one of the things we can actually determine is these 2 parents had to be carriers. They both had 1, and we can determine that they both had exactly 1 copy of the mutant gene. They didn't have 2 copies because they would be affected. They didn't have 0 copies because if they had 0 copies, then all their children would be normal. Even if 1 was a carrier, if 1 had only normal genes, they could only pass a normal gene on, therefore their children wouldn't have it. So we could determine with certainty that the parents had to both be carriers. Male-to-male transmission can occur. Once again, we're going to contrast that with another pedigree later on. Usually involve catalytic proteins, and with the autosomal dominant, we were talking about non-catalytic proteins. The easiest way to visualize that was structural proteins. So usually involve catalytic proteins. What that ends up meaning is, and here's a good general rule to follow, because there's going to be a lot of diseases you guys are going to see throughout the rest of your medical career. If you don't know anything else about the disease, if it's some enzyme involved in metabolism, unless you're told otherwise, it's going to be autosomal recessive. all the metabolic diseases, with very few exceptions, and when there is an exception, it's absolutely pointed out, are autosomal recessive. So almost every disease we talk about in biochemistry in these pathways are autosomal recessive. Occasionally you'll have one that's X-linked, and when it is X-linked, it's pointed out, this is X-linked. Okay? And there are a couple of exceptions where there's some autosomal dominant ones, but those, once again, will be pointed out. So the thing that I always tell people, you know, when you're getting ready for boards and you're studying and you're trying to learn this stuff, just remember which diseases are autosomal dominant and which ones are X-linked, because everything else is recessive, autosomal recessive, and it's a much, much, much bigger list of autosomal recessive. So if you remember the ones that aren't, you know everything else is autosomal recessive. And that really greatly reduces the amount of memorization that you end up having to do, okay? Just some examples that we've seen. So also the metabolic diseases and the hemoglobinopathies. And today we're actually going to talk about one of the hemoglobinopathies, or a class of hemoglobinopathies, the thalassemias. But you've also seen sickle cell already. We talked about cystic fibrosis the last lecture or the lecture before. PKU you've seen. I don't know if you've seen Tay-Sachs yet. If you haven't, you definitely will, but an example of a lysosomal storage disease, all of those lysosomal storage diseases, just like all the glycogen storage diseases, they're all autosomal recessive. There's in a handful of cases you'll find an X-linked one, but unless that's pointed out, then it's autosomal recessive. And so one of the fundamental questions becomes, why is that? So I explained earlier why an autosomal dominant disease, you only need one copy to see the symptoms of the disease. Why in an autosomal recessive disease do you need 2 mutant copies? That's because if you have one normal copy of a gene, it's enough to get the job done, okay? And most of the time we're talking about enzymes and pathways, and if you have 50% of the enzyme, in the vast majority of cases, 50% of the enzyme is good enough to get the job done. It still catalyzes the same reaction, And one thing you can also say is the carriers, in many cases, the carriers we say are asymptomatic. Sometimes if you put them under the right conditions and things get stressful enough on that pathway, they can start to show symptoms. And we'll see examples of that with thalassemias today. But for the most part, having 50% of the enzyme, because that enzyme is normal, is good enough. Another thing also to remember with a lot of these diseases, the disease gene, very often the disease gene is not completely defective. Like doesn't have no activity at all. Occasionally that happens. There are some, definitely, that we've talked about and we will see where that gene is just completely defective, but very often it has some residual activity and is still contributing to the pathway, okay? So sometimes, in many cases, you actually have more than 50% activity in the heterozygote. So just like with the autosomal dominant diseases, I want you to be able to calculate the occurrence or recurrence risk of having a disease. And just like with the autosomal dominant diseases, the one that the diagram showed you was the most common one. This is the most common one that you see with autosomal recessive diseases, which is you have 2 parents who are carriers of a disease. Very often, at first, they don't know they're carriers. But if they had an affected child, then they know they are carriers, and very often what they want to know is, okay, if we have another kid, what's the chance that we're going to have an affected child? And once again, this is a simple Punnett square. I don't want to belabor it. If it's something you haven't seen or you're not getting it from looking at this figure, come see me, and we'll do it in my office on the whiteboard, and once you do it once, you'll know it. It's pretty simple. But we have big A, little a, okay? And remember, when we were talking about the autosomal dominant diseases, the disease gene was the dominant gene, so the big A. But in this case, the dominant gene's always the big A. The recessive gene's always the little a. In this case, The recessive allele is the mutant allele. So what we're looking for is, in this case, somebody with two mutant alleles, little a, to be affected. And if you take two parents who are carriers, okay, big A, little a, and you do this cross, you'll see that 25% approximately, well, they have a 25% chance of having an affected child. Once again, remember, if they've already had two kids and both of them are affected, what's the chance of the next one being affected? It's still 25%, right? So, you know, each one is statistically independent. You could also use this to figure out, okay, what's the chance of them having somebody who's not affected but who is a carrier? Well, you could look at that and figure it out. Well, we got two normal individuals who are carriers in here, so about a 50% chance, approximately 50% chance of them having a child who is a carrier who's unaffected. And they can also have a child, they have a 25% chance of having a child who doesn't have the mutant gene. So here's another couple crosses you guys can do yourself and try to figure out the answers. I didn't give them to you there, but I can give them to you later. Okay. Another thing I can do with pedigrees also is I could give you a pedigree, and one of the things you'll have to do is figure out what pedigree it is, and then maybe I could give you a list of diseases, and you would need to know which diseases would fit that pedigree. So you could work it out and say, okay, this is autosomal recessive, so which of the disease choices did I give you is autosomal recessive? So there are a few key diseases we talked about. The ones that we talked about in this section that I've named for dominant and recessive, you should know. Okay, they're all, I picked only real high yield diseases. Okay, so I'm not asking you to learn any obscure facts. These would be things you would be expected to know anyway. Another thing before I forget to move on, and we're going to get to this by the end of my lectures today. You can see, do you guys remember what the double bar meant? Consanguinity or incestuous mating, right? So we got some cousins here who are getting frisky and apparently very frisky too, because they had a lot of kids. So anytime you see the double bars in a pedigree, almost without exception, that's a clue that they're talking about autosomal recessive. Okay, and we're going to talk about later on why that is, but unless you have a compelling reason to think it's something else, when you see the double bars, the first thing that should pop into your mind is we're looking at an autosomal recessive pedigree. Okay, but you know, once I guess the COMLEX might try to throw a monkey wrench in there, but if you're familiar with the pedigrees, you could figure it out. But you should be thinking that. Okay. So here's some other things I could ask you with the pedigree. What is the genotype of the parents in the third row? So I don't have the rows labeled, but 1, 2, 3. So these two cousins who are having a large family, what is the genotype? So it could be any one of these three. Let's figure out which it is. Big A, big A. Well, that's a possibility at first, right? Because they're not affected. So we know it's not little a, little a. We can eliminate that right away because they're not affected. Okay, so it's got to be one of the two. Okay, so they're having affected children, though. So that means that each one of them needed to pass on a mutant recessive allele. That means each of the parents both had to be big A, little a. They're both obligate carriers in this pedigree. Okay? Another question I could ask, what is the genotype of the unaffected children? Okay, so we know what the genotype of the affected children is, right? Big A, little a. So we have two choices of what the genotype of the children could be. It could be big A, or big A, little a. Oh, I threw a monkey wrench there. I asked you a question, and there was actually two possible answers there. You can't determine with 100% certainty without doing a molecular test here what the genotype is. Because each of the normal children, they could either be big A, or they could be big A, little a. They could be carriers. Remember, we did the pedigree earlier where you did that cross. So in that case, that was a little trick I threw in there. But you guys should be able to figure that stuff out. Now, I'm not going to give you a question on an exam where there's 2 correct choices, at least not on purpose. Okay, okay, so one of the diseases that I introduced in this section, and it's always something that I always include in genetics. In every genetics text, always devotes a large section to the thalassemias. Now, have you guys in med school here seen thalassemias yet? You have? Oh, awesome. Which class did you see it in? Okay, so he talked about it during the hemoglobin stuff. Okay, so good. I'm probably going to go into maybe a little bit more detail than he did, so we're going to spend a little bit of time on the thalassemias. This is one of those things that's worldwide so common, such a common genetic disease that you guys pretty much need to know this inside and out. Okay, so you need to know all about the thalassemias. And the genetic stuff in the thalassemias becomes, at least for a nerd, extremely interesting. Okay, so first of all, thalassemias are autosomal recessive diseases. Okay, we classified that in the autosomal recessive diseases. And what a thalassemia is an imbalance in globin chain synthesis. So one of the globin chains is you're not producing enough normal of one of the globin chains, and then that results in the symptoms that we're going to talk about. If you group them together, they're the most common single gene disorders in the world. So if you group all the alpha and beta thalassemias together, that's why it's such an important topic. Thalassa means sea, so and that's where this disease was originally characterized, was around the Mediterranean Sea, and that's where the thalassemia came from. But you will also see it in parts of Africa, Southeast Asia. One thing I will tell you is that the places that have the highest incidence of thalassemias are also the places that have the highest incidence of malaria. That's something we'll touch on in the next lecture. 2 types of thalassemia. We have our alpha thalassemias and our beta thalassemias. So the name of the thalassemia is whatever chain is deficient. So for alpha thalassemia, the alpha chain is deficient, and beta thalassemia, the beta chain is deficient. However, one thing to remember is when one chain is deficient, the other is in excess as well. And in the thalassemias, both the chain that's deficient, okay, and that's going to cause, when you have not enough hemoglobin, that automatically causes an anemia, right? In this case, the excess chain also contributes to the problems. Okay, so the excess chain is going to accumulate and contribute to the symptoms of the disease, as we're going to see. So some basics about hemoglobin, and I guess for the most part this should be a reminder. But this needs to be fresh in your head when we go through how the symptoms are going to manifest in the thalassemias. So you guys remember adult hemoglobin, you got 2 alpha chains and 2 beta chains, right? And you may see me use this abbreviation, HbA. There's going to be several unique hemoglobin types that we're going to talk about, and you should be familiar with those. We'll talk about HbA, HbH, we'll talk about fetal hemoglobin, talk about hemoglobin BART, and so we're going to talk about these different hemoglobin, and you should be comfortable with those. As far as the genes go, and this is kind of one of the places it's kind of interesting, there's a single gene on chromosome 11 for the beta globin chain. You have 2 chromosome 11s, that means you have 2 beta globin genes. However, for the alpha globin gene, you have 2 it's actually chromosome 16, you have 2 right next to each other on chromosome 16. You have 2 of chromosome 16, so you actually have 4 in a normal situation, 4 alpha chains. Okay? Because those genes are right next to each other on the chromosome, it actually creates an opportunity for things to go wrong during recombination. So we're going to talk about the alpha thalassemias and some missing genes shortly. Okay, in order to understand what's going on with thalassemia and how the disease is going to present, you also have to understand a little bit about fetal hemoglobin. So in fetal hemoglobin, you have these gamma chains that replace the beta chains. Okay, so fetal hemoglobin has alpha 2, gamma 2 instead of alpha 2, beta 2. And that's because that hemoglobin, that fetal hemoglobin, or HbF, fetal hemoglobin, it has to be able to get oxygen from the mother, who already has normal hemoglobin, HbA, right? So the HbF has to be a little bit different. It actually has to have a little bit higher affinity to be able to pull oxygen from the mother. So that's the difference with fetal hemoglobin. It's got a higher affinity for oxygen. Now, it's still within the range where it can still deliver oxygen to tissues, but it's a little bit higher affinity, okay? Now, what happens is, so in utero, the fetus is producing, at least in the simplest model, and let's just keep it simple, it's just producing 100% hemoglobin, fetal hemoglobin, hemoglobin F. After birth, what happens is that hemoglobin F, or the synthesis of these gamma chains, starts to go down. So over the course of about 6 months, it goes from 100% down to 0. And simultaneously during that time, the beta chain synthesis goes up. Okay, it starts at 0% and then goes up to 100%. Okay, and that process takes about 6 months. Okay, now I put a little star here because I wanted to point out, I'm talking in absolutes to make the model very simple so you guys grasp it the first time. You do have a little bit of fetal hemoglobin. You can measure fetal hemoglobin in adults. There is a small amount there. So I don't want you to, later on you may see some lab values and find out, oh wait, what's going on here? I'm seeing fetal hemoglobin. Maybe there's something weird going on. You do have a little bit, okay? But it's easier just to think 0 and 100 and changing over time. So that's going to affect the way these diseases manifest, and so we'll talk about that shortly. Okay, so first we're going to talk about the beta thalassemias. In the beta thalassemias, once again, the beta globin chain is not being synthesized in sufficient amounts. Okay, at least normal beta globin is not being synthesized in sufficient amounts. This means that the alpha globin chain is in excess, and that excess of alpha globin chains actually accumulates as a tetramer. You can almost think of it as being a pseudo hemoglobin molecule. If there's not enough beta globin to pair with, it tries to pair with itself. Okay? However, these alpha globin chain tetramers are very insoluble. Okay? And you've already seen examples of other proteins where you have something that's very insoluble. Insoluble, it precipitates out, it ends up killing cells, okay? And that results in a hemolysis, so a hemolytic anemia. So with the beta thalassemias, you got a double whammy here. You don't have enough beta chain, okay? So that's already causing an issue. That's already causing an anemia. In addition, the alpha chain tetramers that accumulate are also toxic, okay? And so they precipitate out and they cause red cell turnover and a hemolytic anemia. So that contributes to the symptoms. Both of those contribute to the symptoms, okay? So with our beta thalassemia, we're going to start out with the simple model. So we said it was an autosomal recessive disease. That means you can have 2 normal genes, 1 normal gene and 1 mutated gene, which should be an asymptomatic carrier, right? And then 2 mutated genes. One thing I want to point out is, because I got this from your textbook, and I wish they used a little bit different figures, and maybe I should look in the latest edition and see if they've changed it, but I don't think they have. Because with the alpha globin figure, it's going to look very similar. In this case, beta globin mutations are mutations. When we get to the alpha globin genes, they're actually missing genes. And I want you to recognize that. So it looks like the gene is missing here, but it's some kind of mutation. Okay? And we'll talk about the different types of mutations. So in the simplest scenario, you can have 2 normal copies. You could have 1 normal copy and 1 mutant copy, which should be asymptomatic, right? if it's an autosomal recessive disease. That actually causes beta thalassemia minor, which I'm going to get to in a second. And then you can have two mutated copies, which is the major disease, and it's beta thalassemia major. It's also very often just called thalassemia major. When you hear thalassemia major, they're talking, or thalassemia minor, those terms are used for the beta thalassemia versions, okay? So the alpha thalassemias, when we get to it, I'll show you, they have a little bit different names. Okay, so I guess one thing I should point out at this point is, now I just said we're talking about an autosomal recessive disease, and I just gave a condition name for having one mutated copy. If it's autosomal recessive, the carrier should be normal, right? And actually, people who are carriers for beta thalassemia, for the most part, are normal. But this is also where sometimes the line between what's called autosomal dominant and what's called autosomal recessive starts to get a little bit blurred. Okay, I've already showed you where it gets blurred when we talked about autosomal dominant. If you get two mutant autosomal dominant genes, the disease phenotype is even more severe. So then why is it maybe it should, maybe how do you say that that's dominant? So your textbook, if you want to read a little bit about that, your textbook has a little discussion on that, and where the lines start to get a little bit blurred between recessive and dominant. For the most part, though, the hemoglobinopathies, when you just have, and you see the same thing with sickle cell, right? You've already talked about sickle cell. You get one mutated copy of the sickle cell gene. For the most part, you're normal, but under certain conditions, you can start to see some symptoms. So the line gets a little bit blurred there. Okay, so I used the beta thalassemia in an earlier lecture when we were talking about mutation types. So you've already seen this particular figure. So with the beta thalassemia, there's a large number of known mutation types. I showed it to you earlier as an example of a disease where very frequently we see splice site mutations, and those are the little dashed arrows there. Those are splicing defects. Okay. I also used it as another example, or as the example of a disease where you could possibly have a promoter mutation. Okay. There's also where you can have sort of your classic nonsense or missense mutations. So a bunch of different known mutations with the beta thalassemia. Now, some of these mutations are going to absolutely, completely knock the gene out in terms of its function. Something will still be produced there, but it won't be normal and it won't be able to function normally. Some of these splicing defects, it could either completely knock it out because you never produce a normal one, or it may affect the amount of normal you produce versus non-normal. Or if you had a nonsense mutation in there, you'd never produce any normal beta globin from that gene. But then you can have things like promoter mutations where everything you produce from there would be absolutely normal, but you're just not making enough of it. Okay? So that kind of starts complicating the picture of what's going on with the beta thalassemias. And that means we can have some mutations that essentially knock the gene out in terms of its function. It's not missing like in the alpha thalassemias, as we're going to talk about. It's there, but it's not producing any functional protein. And that's a beta 0 or a beta null gene. Whereas some of them, and I think the easiest way to think about this, some of them can produce normal beta globin, but just in reduced amounts. And I think the easiest place to think about that is the promoter mutations. Okay, what does get produced is normal. You're just not making enough of it because the promoter's not working efficiently. Okay, so unfortunately, what that means is now the way beta thalassemia presents can start to get very complicated, okay? Because you can have two completely normal genes, two completely, essentially for all intents and purposes, two genes producing absolutely no normal beta globin, But then you can have everything in between as well, because you've got all these different types of possible beta plus mutations. So you can get different amounts of normal synthesis, and that creates this wide spectrum of how beta thalassemia actually presents. Okay, so the simplest model is you got your minor and major. In reality, there's a whole spectrum, and there's an additional phenotype included in there called beta thalassemia intermedia, and it's somewhere between minor and major. And I'm going to define those for you right now. So beta thalassemia minor, in many cases they are completely asymptomatic, just like people who are carriers for sickle cell. In many cases they will never even know they're carriers for sickle cell, but under the right conditions they may show some symptoms. They may have a mild anemia, in particular under conditions where anemia might be induced. Okay, they might be more likely to show anemia than an average individual. It's very easy to mistake this for iron deficiency anemia. By the way, anemia is one of those things that's absolutely enormously huge on the boards. Luckily, we have somebody here who is a real expert on that, so Dr. Siddiqui will tell you everything you ever need to know about anemia, so I'm confident you guys are going to be experts going in. But just as an aside, because I'm sure this will be driven in later, the iron deficiency anemia is the most common one. Okay, so that's always the first suspicion, and so that's and it can be confused with iron deficiency anemia. So you have to go into some little bit deeper tests to differentiate that. Hemoglobin electrophoresis, so certainly you can find those alpha chain tetramers, and some blood work, and I'll let the hematologist discuss some of the blood work in later classes that would be done. Okay, so that's the minor version, and then you can have the major version. In the major version, you either have no normal beta globin being produced or very small amounts of normal beta globin being produced. That's going to cause the major symptoms. Now, in order to understand how, and so we're going to talk about what the symptoms are besides just the anemia. In order to understand how the symptoms manifest, you have to go back and recall what's going on with fetal hemoglobin, right? In fetal hemoglobin, that beta globin is not there, right? So if you have a baby born with thalassemia major, okay, they are going to appear at birth in utero. They're going to be absolutely, completely fine, absolutely, completely normal. And what's going to happen is after they're born, and at first after they're born, they're going to be completely normal as well. But what's going to happen is once that fetal hemoglobin gene starts decreasing the amount of fetal hemoglobin, or the gamma globin gene starts to be shut off, and the beta globin gene starts to be turned on, that's when the symptoms are going to start to manifest. So over a relatively short period of time, the symptoms are going to get more and more severe. So you're going to have a baby that appears perfectly normal at birth. The parents are all really happy we've got a normal baby, and then over a period of time they drastically decline. Okay. So does that make sense to you? What's going on? So that's important to recognize with beta thalassemia. Be asymptomatic at birth, and then it's going to get a rapid decline as the beta globin gene synthesis goes up. What are going to be the symptoms? You're going to have a very severe anemia. In thalassemia minor, you've got a very mild anemia or no anemia. Thalassemia major, you have a severe anemia. So you both have deficient hemoglobin. Okay, you don't have enough beta globin there, or maybe no beta globin there, so you're not making normal hemoglobin, or certainly not enough of it. That by itself is an anemia. In addition, the alpha chain tetramers are going to accumulate, and the accumulation of those alpha chain tetramers is going to cause a hemolytic anemia as well. Okay, they precipitate out; those cells get get recognized as being damaged, and they're pulled out of circulation. Okay? So you get the symptoms of a severe anemia, and then you also get some additional symptoms we're going to talk about right here. So you get an enlarged liver and spleen. So what's happening here is your body is attempting to increase the red cell production. Okay? It recognizes there's an anemia going on. We don't have enough red blood cells to carry out normal function. So what it does is it goes into overdrive. And actually, normally your liver and spleen, under normal conditions, aren't heavily involved in producing red blood cells. It's just when you're in a crisis like that, they do have the ability to produce red blood cells. So they actually kick on. And that's actually going to cause an enlargement of the liver and spleen, which I'll show you in 1 second. Another thing that you'll see in untreated beta thalassemia major, or thalassemia major, is skeletal deformities, very, very common. Particularly noticeable skeletal deformities will be seen in the face and the skull. So I'll give you an example of a picture in a second, but you can type this into Google Images and find some more pictures as well. And this is the expansion of bone marrow. Once again, the body's reacting. It's saying, okay, we've got a severe anemia here. We need to increase the amount of red blood cells we're producing. Unfortunately, that's not going to solve the problem here, but that's what's going on and causing those symptoms. Another thing you see in patients with thalassemia major is systemic iron overload, okay? So, and this is due to the red blood cell turnover. We get this hemolytic anemia. You're constantly turning over red blood cells. And when you do, you turn over the iron in that cell. Your body recognizes that you're anemic. So your body reacts to anemia by trying to pull in more iron. Okay? That's how your body reacts. And you'll talk about iron absorption later on if you haven't already. But it's going to react by trying to get more and more. So it's actually going to start accumulating iron, and you get systemic iron overload. So it accumulates in the liver and heart. So that's also a symptom of thalassemia major. Here's a couple pictures of what you see with the skeletal deformities. So under an X-ray, you can actually see that's not hair right there. That's actually the skeletal deformity going on in this individual. You see the massive hepatosplenomegaly here, massive liver and spleen. And this is in an untreated individual. Once again, you can find tons of pictures of this online. So can you actually treat thalassemia major? Yes, there are some treatment options available. One thing that would be required is a regular blood transfusion, okay, to give them some normal red blood cells. You have to couple that with iron chelation therapy. It's not unusual, or when you're giving people regular blood transfusions anyway, you have to do iron chelation therapy because you tend to overload them with iron with all the extra blood you're giving. But in addition, with the thalassemias anyway, they're already overloaded with iron. So it's really important to have that iron chelation therapy to reduce the potentially fatal consequences of having too much iron as well. You can do a bone marrow transplant, and that could be potentially curative if you can find a suitable donor. So if you can find a matching donor, now bone marrow transplants are very dangerous. So and once again, it's hard to find a suitable donor, but that could potentially be curative. Okay, so that's and there are gene therapy options that are being looked at. We're going to talk, Dr. Gardner will talk to you a little bit about gene therapy. I don't know if you'll mention beta thalassemia in there, but after you learn a little bit about gene therapy, you'll be able to very quickly figure out how you could use it for beta thalassemia. And there's actually some very aggressive genetic screening programs in some countries that have really, in countries where there's a high prevalence of thalassemia, that have really reduced the incidence of this disease. Okay? Thalassemias aren't the only only diseases where you have this. You see it also with Tay-Sachs in certain populations, but the screening has helped. So that reduced the incidence a little bit. Now, I told you we have this spectrum of symptoms from major and minor. There's also a thalassemia intermedia that confuses the picture a little bit. So remember, this is a clinical term, so it's not determined absolutely by molecular genetics. So they don't go in, sequence the genes and say, okay, you have intermedia, or sequence the genes and say you have major. They do it based on symptoms. So, and we're going to talk about some genetic diseases where it's not based on the molecular defect, but it's based on how the symptoms present. Okay, so that's a clinical terminology, and there can be a wide spectrum between major and minor. Okay, so how do we define intermedia then versus minor and major? Okay, so basically minor requires no blood transfusions. Major requires transfusions on a regularly scheduled basis all the time. And intermediate does require blood transfusions, but they don't have to be done on a regular basis. So how we determine how often, what they do is they'll monitor those patients, and when their hemoglobin levels get to a point where the clinician feels they need a transfusion, they'll do it, but it's not regular, okay? So, and people always ask me, what do you mean by regular? So I went and asked a clinician because I didn't know and it wasn't defined in the book. I'm like, what do they mean when they say regular transfusions? Somebody who's undergoing regular blood transfusions, what I was told was every 2 to 4 weeks would be considered a regular blood transfusion. With intermedia, it would only be as necessary, okay? So that's basically how they define those. Here's a good question for you. What if the gamma globin synthesis persisted into adulthood? Well, if it persisted into adulthood, then you would have fetal hemoglobin around all the time, right? Now, fetal hemoglobin still works. It doesn't work as good, meaning it binds a little more tightly so it doesn't release oxygen as good, but you can still survive with fetal hemoglobin. And actually, there's a condition called hereditary persistence of fetal hemoglobin, where that actually happens. And those individuals, for the most part, are normal. Probably never going to be a world-class athlete, but they're going to be normal in terms of a normal lifespan. So if you can figure out how to turn on the gamma globin gene, you could actually potentially cure diseases where you have a defect in that, including beta thalassemia, including sickle cell anemia. And actually, that's what, I don't know if, did you guys hear about hydroxyurea being used in sickle cell? Okay, so that's what hydroxyurea does. It turns on this fetal hemoglobin gene. So that's something that's definitely being looked at as well. So with our alpha thalassemias, the situation is a little different. With alpha thalassemias, we have very defined genotypes because we don't have these intermediate genotypes. What version of alpha thalassemia you have depends on how many genes you have. So in alpha thalassemia, by far, what causes this in most of the cases is missing genes. That's because you got these two genes next to each other. They look very similar, so during recombination, they can recombine ineffectively, where one chromosome ends up with 0 and one ends up with more. Okay, so or one chromosome ends up with 1 and one ends up with 3, I should say. Or eventually you could end up with 0, and you could have 0 and 4. You could have all these different possible combinations. Ultimately, actually having too many for the alpha globin gene is not really an issue, but if you don't have enough, it can become an issue. And so there's 6 possible genotypes. We can either have 4 normal genes, or we can have 1 missing gene, 2 missing genes, 3 missing genes, or all 4 missing genes. Okay, and each one of those has a very specific name. They're either normal, silent carrier. Okay, they have 1 mutated gene. Alpha thalassemia traits, when you have 2 mutated genes, one of the things to notice about that is in this case, the 2 mutated genes, or the 2 missing genes, I should say, the 2 missing genes could come both from 1 chromosome, or you could be missing a gene on each of your chromosomes. Okay, so that's why you have this "and" in there. Could be either possible scenario. which can make a difference if these people were to go to have kids themselves and who they mate with. HBH disease is when you only have one normal copy, and then finally hydrops fatalis is when you have no normal copies. Hydrops fatalis is not only due to alpha globin or alpha thalassemia. That's a general term that you see. There's other conditions that cause hydrops fatalis. Okay, here's a table for you guys. I believe I actually got this out of Robbins Pathology, and I pulled it out of an older version, but I think the new version is exactly the same as this. But I love this table because it tells you everything you need to know about thalassemias, except what you can do is you can block the genotype out for the beta thalassemias. That gets too confusing because of the range of different genotypes. So if you black that part out, everything you need to know about thalassemias is in there, all summarized in one nice, neat little table. Okay, that's really cool. At least to me, it's really cool. Hopefully you'll find it really cool because it makes it easy to study. Okay. So once again, you got your silent carriers. They got 3 normal genes. You got the people who have 2 normal genes and 2 missing genes. Generally, they are asymptomatic or may present like thalassemia minor, a very minor anemia. HBH disease, you only have 1 normal copy. Okay. And they're actually going to have the symptoms very similar to beta thalassemia intermedia. So it's going to be a mild anemia. That's what beta thalassemia intermedia is. So you're missing 3 genes. You only have 1 gene. You should have 4. You're missing 3, but yet you're only getting a mild anemia. That's, to me, sticks out as being a little bit odd, and I want to explain why that is. However, you do have to have at least 1 normal copy, because if all 4 are missing, you get this hydrops fatalis. You get death in utero or very shortly after birth. Okay, so this is not compatible with life. You have to have at least 1 normal gene. So we talked a little bit about fetal hemoglobin earlier. So in fetal hemoglobin, you have that gamma globin chain substituting for the beta globin. And what you'll see in patients with alpha thalassemia is this gamma globin in utero or shortly after birth, while it's still present, will accumulate as a tetramer called hemoglobin BART. Once again, it's kind of a pseudo hemoglobin molecule, but it's unique. Hemoglobin BART is unique to alpha thalassemia, and it's a tetramer of that gamma globin chain that's present in fetuses and shortly after birth. Unique hemoglobin that you'll see there. Now, eventually, when the beta globin chain synthesis takes over, that hemoglobin BART is going to go away, and you're going to get the beta globin tetramer accumulating, and that's going to be, that's actually got a name as well. It's called HBH. Okay? We didn't have a special name for the alpha chain tetramer. It's just called the alpha chain tetramer. But we do have a special name for HBH, or for the beta globin tetramer. Now, both HBH and hemoglobin BART are actually more soluble than the alpha chain tetramer. And that's why, because they're a little bit more soluble, they still cause issues. That tetramer still causes issues, but it's not quite as severe as what you see in the alpha chain tetramer in the beta thalassemias. Okay? So what ends up happening is the symptoms tend to be a little bit less severe in the alpha thalassemia. So the beta thalassemias with a similar amount of chain imbalance have a more severe phenotype than the alpha thalassemias, and that's due to the tetramer that's accumulating being a little bit less soluble. And there, I've given you a list of all the different types of thalassemias, so that was just for your study purposes. Okay, so the next thing we're going to do is the Hardy-Weinberg equilibrium. I'm going to get this started. I'll probably have to finish it in the next lecture, but I'm going to go over a little bit with you of this. So in order to use so this is going to be some population genetics and some math we're going to use, and I'm going to show you there's a specific purpose, and it's actually kind of a neat and interesting purpose we use it for. But in order to use it, you have to make some assumptions. The population is large, and individuals made at random. You know that's not always true, right? Individuals don't just go around mating at random, at least not normally. And you know, they're also making some other assumptions there that their genes aren't coming in and genes aren't leaving and things like that. But if you assume all those things, you can make a constant and predictable relationship. So we can use gene frequencies to determine how often you will see a disease, or if you know how often you see a disease, you can determine gene frequencies. And the real thing we're actually going to end up using it for is we can actually determine, ultimately we can determine the carrier rate for autosomal recessive diseases, which is kind of neat. And it'll actually, to me, it's amazing when you take a real rare disease and then you see how common that the gene can be in the population. When we're applying the Hardy-Weinberg equilibrium, we're always talking just about two alleles. So we'll have a normal allele and a mutant allele. We're always talking about an autosomal recessive disease. Okay, so you can assign either P and Q, the normal or mutant allele. They have to add up to 100%. That's going to be 100% of the gene. So if you know the percentage of one, then you automatically know the other, right? That's all this math right here was trying to show you. You know one, then you know the other, okay? And then so we get this equation, and I guess, did I write the equation out? So you get P squared plus 2PQ plus Q squared. That's going to be our equation. And that's going to tell us, if we know the gene frequencies, how often you see people with 2 Qs, which is Q squared. How often you see people who are heterozygotes, that's 2PQ. Or how often you have people with 2 P genes, which would be P squared. Okay, so you could just plug those numbers in. Okay, and I've done a little bit of simple math for you guys right there. And you could, verify the math yourself. It's pretty straightforward and pretty simple. And they have to add up to 1, right? The frequency of those genotypes, you only have 2 possibility of different genes, are all going to add up to 1 at the end. Okay, so what we're ultimately going to use this for is to determine the incidence of an autosomal recessive disease, okay? So I'm going to, just so we're all fresh in our heads, I'm going to delay this part and put this in the very beginning of the next lecture so you guys have a chance to have a break, okay?